Hello, this is the oral portion of the first exam in mammalogy. So to begin, we're going to first talk about the three important evolutionary transformations from non-mammalian animals to mammalian animals. One of them being the secondary palate. This shunts air from the front to the back of the mouth, and this enables mammals to breathe and eat at the same time, and specifically suckle and breathe at the same time, which is very, very important for mammals. Another transformation would be the parasagittal plane. So previously, mostly seen in reptiles, their limbs are to the sides of their body or more sprawling. And this parasagittal plane enables mammals to have vertical movement below the body and is less limiting than as the sprawling that we see in reptiles. And another evolutionary transformation would be the hammer and anvil. These were once jaw bones in reptilian ancestors, and these jaw bones eventually migrated to what is now the middle ear in mammals and is thought to have improved hearing in the early mammals. And moving on to the three subclasses of mammals, we first have the monotremes. Two examples of these are the echidna and the duck-billed platypus. And monotremes diverged in the late Jurassic and early Cretaceous periods, and their main stronghold is in Australasia. In fact, you can only find these guys in that region. They have reduced dentition or just lack teeth altogether. They have limited running ability. They do have a cloaca, so very similar to our birds and reptiles, which is very interesting. And they also have mechanoreceptors and electroreceptors on their bill or beak, very similar to the ampullae of Lorenzini in sharks. And for reproduction, they are oviparous or egg-laying mammals. They also lack teats, so very interesting. The echidnas, they don't have teats, so instead of the young suckling, the young will kind of just lap up the milk from the mother, which is very interesting. They also develop a temporary pouch when they're developing the young as well. Then we have the marsupials, who diverged in the late Cretaceous periods. They have strongholds in Australasia and the neotropical regions. Two examples of marsupials would be the koala and the Virginia opossum, which is my personal favorite. They only have one set of teeth. They also have a cloaca like monotremes, but they have a lower body temperature and a lower basal metabolic rate compared to placentals. So their body temperature is around 35 degrees Celsius. And for reproduction, they have very underdeveloped young because they have such a short gestation period. These guys, however, do have a pouch that houses the young as they're developing. And then there's the placental mammals, who also diverged in the late Cretaceous. They're found almost everywhere except for Antarctica, two of them being the river otter and the tiger. So placentals have a higher body temperature than marsupials. Their temperature is around 38 degrees Celsius. And we have less teeth than marsupials, but we have two kinds. So we have those baby teeth and then the more permanent teeth that come in later on. For reproduction, placentals have more precocial or more developed young because of that longer gestation period, and placentals do not have a pouch. And I chose smell for one mammalian sense organ. Specifically, I'm going to talk about the vomeronasal organ. It is the largest gene in the genome, and the vomeronasal organ can detect pheromones and different kinds of scents. We see this in a variety of animals, including snakes and felines as well, which we'll talk about in just a moment. And this vomeronasal organ is very acute and specialized in mammals. This could possibly be linked to the nocturnal origin of the early mammals. So as I was saying, the vomeronasal organ, or the Jacobson's organ, is seen in a variety of animals, but they kind of work in different ways. So specifically for the snake and some lizard species as well, they will stick out that forked tongue of theirs and pick up the scent particles with that tongue and kind of bring it back into their mouth and stick the tongue up into the Jacobson's organ, which is basically just two holes on the roof of their mouth. And that's how they're able to sense their environment and be able to smell very well. In felines, it's a little bit different. They don't have that forked tongue to pick up the scent particles. Instead, they'll kind of just scrunch up their face really tight, and they're able to kind of inhale all of those scents and those pheromones, which is very interesting to see. And timing of reproduction is very important for mammals, and different mammals go through different periods of reproduction. So one of them being delayed fertilization, in which the female will kind of store the sperm until the environmental conditions improve, and then the sperm will be fertilized. And we can see that in the European hare, which is very interesting. There's also delayed implantation, which is where further development of the blastocyst is stalled. So fertilization has occurred, but implantation in the uterus has not. And we can see that in kangaroos. It's very interesting because the mother can have a joey that is just weaning off of milk, so he's out of the pouch but still kind of suckling the milk a little bit. She can have a young joey who is still developing in the pouch, still attached to that teat. And then she can also have a blastocyst in her uterus as well. So she can pretty much have 3 babies at one time, which is very, very interesting, and it takes a lot of energy to do that. There's also delayed development, which is seen in the leaf-nosed bat, and that's where the blastocyst implants in the uterine lining, and because of that, it stalls any further development. And this can delay parturition until food availability is the greatest. So it's better to, raise babies when there's a lot of food than, when there's not as much food available. And specifically for bats, this could be useful because bats are colonial. This can cause synchronous parturition, which is kind of beneficial for all of the bats in the colony. And so for hibernation, besides the Arctic ground squirrel that goes through deep hibernation, some bats can also go through this. And so I chose the little brown bat, or Myotis lucifagus. So they inhabit the forests of North America. So they can live in the United States up into Canada and also parts of Central America as well. And they enter this hibernation around September, October. It depends on the gender. So I believe the females will enter hibernation earlier than the males will. And they emerge from hibernation in April or May. And they do this in caves or abandoned mines. And so what's really interesting is very similar to the ground squirrel. They will severely reduce their metabolic rate and their heart rate. Their breathing slows down to a minimum. So these bats can reduce their metabolic rate to about 1% of their resting metabolic rate. And their heartbeat drops to about 20 beats per minute, where normally it's 210 beats per minute. So they kind of slow down their whole entire body. And of course, they emerge in the springtime. So they go through this hibernation to save energy, to be able to live through the severe environmental conditions that these bats can live in, depending on where they are in North America. And of course, they go through the spring migration right after they come out of this hibernation period as well. And finally, we're going to go over the two diseases found in deer that Dr. Brown gave his lecture on. So the first one was hemorrhagic disease in deer. This affects wild and some domestic ruminants. And the vector are these coolicoids or biting mites. And this disease attacks the blood vessels and progresses, eventually leading to ulceration and death ultimately. And this has a seasonal peak as well. So as the temperatures warm, we get more of those biting mites, and that's where we see this disease kind of spike in the deer population. It also depends on the region as well. So if you're far more south, this disease occurs more frequently, but it's less severe in the population. Whereas if you go farther north, this disease is less frequent. However, it's more severe. It's going to affect the deer population a lot more than in the southern areas of the US. And because of this, there's really no management strategies. It's kind of like, we'll take it as we come sort of thing. There's no real treatment for it. It's kind of just dealing with it each and every year, depending on where you are in the US. And then the second one is chronic wasting disease. This affects cervids such as elk, deer, and moose. And this is caused by prions, which is an abnormal form of a protein. You can see it in the picture to the side there. And this disease attacks the brain and the nervous system and ultimately leads to death. And unfortunately, there's no known treatment or vaccine. So again, there's really no specific management strategies or treatment options, unfortunately, which is very, very sad. So that was my oral presentation for the first exam. Hope you enjoyed.